In recent years, many basic studies on iPS cells have been conducted in veterinary regenerative medicine. Let me introduce our research work here first. In order to establish clinical-grade canine iPS cells, we have generated canine iPS cells without using viral vectors. Furthermore, we have identified a method for culturing iPS cells without feeder cells. To use iPS cells in clinical practice and for the development of animal regenerative medicine products, it is believed to establish iPS cells from somatic cells that can be harvested without invasive procedures on the animal. Therefore, we focused on biowaste such as canine umbilical cord-derived tissue and attempted to generate canine umbilical cord-derived iPS cells. As a result, we succeeded in generating canine umbilical cord-derived iPS cells without using viral vectors. Numerous studies have been conducted for the clinical application of canine iPS cells, and many papers have been reported so far. However, there has been little research on the establishment of feline iPS cells, so this will lead to my main presentation today. In 2012, it was reported that iPS cells in snow leopards were successfully established. This study used fibroblasts of snow leopards as a somatic cell source, and 5 reprogramming factors were transfected using retroviral vectors. As a result, they have succeeded in establishing iPS cells that have been confirmed to express stem cell markers and to have teratoma-forming ability. So you can say that the feline iPS cells were established for the first time in the world back in 2012. As for domestic cats, the iPS cells were established for the first time not until in 2019. This study used adipose tissue-derived fibroblasts as a somatic cell source, and 5 reprogramming factors were transfected using retroviral vectors as well, and they have succeeded in establishing iPS cells of domestic cats. In this study, they have revealed that the culture of these feline iPS cells needed FBS and feeder cells. Also, they have been confirmed to express stem cell markers and to have the ability of taurinase differentiation in these feline iPS cells. Nevertheless, there have been no reports of the generation of feline iPS cells without using viral vectors and feeder cells. Our objective was to generate feline iPS cells without using viral vectors and feeder cells. In order to do that, we collected ovaries and uteruses to derive fibroblasts and use them as somatic cell source for the iPS cells. We used the ovaries and uteruses discarded during the ovarian hysterectomy of cats in this study. First, whole tissue was washed, and the ovaries and the uterus broad ligaments and uterine horn were separated. These separated tissues were cultured with DMEM plus 10% FBS. As a result, fibroblast-like cells were successfully derived from the ovary, uterine broad ligament, and uterine horn. Among these cells, ovary and uterine broad ligament-derived cells exhibit a high proliferation rate. Hence, we used these 2 cell lines to generate feline iPS cells. Next, episomal vectors carrying the reprogramming factors were transfected into fibroblast-like cells derived from the ovary and uterine broad ligament. The reprogramming factors have succeeded in establishing canine iPS cells were used in this study. After transfection of the reprogramming factors, we attempted to establish iPS cells using 2 types of ESL medium. When putative iPS cell colonies formed, they were extracted and cultured to analyze the characteristics of the iPS cells. After expanding the iPS cell colonies, we confirmed morphology and proliferation capacity, the expression of ALP and stem cell markers, and taurinase differentiation ability. After the transfection, the efficiency of electroporation was found to be about 20% in both cell types. When the post-transfected cells were cultured in naive ESL medium, multiple primary colonies emerged around day 10 after the transfection. These cells formed colonies even after passaging to new feeder cells and changing the medium to primed ESL medium. However, the colony morphology of the iPS cells could not be maintained in primed ESL medium. We observed that it's difficult to maintain the putative iPS cells in primed ESL medium, but the iPS cells could be maintained in stem feed AK02. Also, we observed that these iPS cells grew well and formed colonies on the I-matrix coating. Initially, there were many differentiated cells, but the number of differentiated cells tended to be eliminated after each passage. A magnified image of a feline iPS cell colony was shown in this slide. The cells of colony was observed to be spherical cells exhibiting a high nuclear cytoplasm ratio specific to pluripotent stem cells. This colony morphology was slightly different from that of human iPS cells. Also, we demonstrated that the iPS cells maintain high proliferative potential for at least 10 passages. To test pluripotent characterization, we stained feline iPS cells with LP staining, and the undifferentiated colony was positive. When the mRNA expressions for stem cell markers were evaluated, the mRNA expressions of feline and genus OCT4 nano-SOX2 in the iPS cells were higher than those of fibroblast-like cells. And we found that immunofluorescence staining of stem cell markers in these iPS cells were positive for OCT4, SOX2, NANOG, and SSE1. We also demonstrated that embryonic bodies from the iPS cells differentiated into all three germ layers. It is interesting that the colony morphology of the feline iPS cells in this study was a mixture of dome-shaped and flat-type colonies, indicating that they may be intermediate between mouse ES cells and human iPS cells. In addition, the expression status of stem cell markers was found to be identical to that of mouse ES cells. In this study, we succeeded in generating feline iPS cells on feeder cells by introducing reprogramming factors using episomal vectors. And we confirmed that feline iPS cells can be maintained under feeder-free conditions. However, optimal conditions for the generation and maintenance of feline iPS cells might be different from those for the culture of canine iPS cells. From now on, the optimal conditions for the generation and maintenance of feline iPS cells needed to be further adjusted. Also, karyotyping and confirmation of teratoma-forming ability are still required. In this study, we succeeded in generating virus-free and feeder-free feline iPS cells, and these cells may be one of the leading candidates for realizing regenerative medicine in cats. In conclusion, we have succeeded in generating feline iPS cells without using viral vectors and feeder cells. Thank you for your attention.
Thank you so much.
Thank you.
Any questions?
Thank you very much, Atsushi. It was a great presentation. I have lots of questions, so I'll ask one and then we'll see if other people have other questions. So the factors that you used for transfection, so it seems like the there was quite a few of them, definitely more than the 4 Yamanaka factors and the 2 other, Nog and Lin. You had a few other ones there. So how did you come up with this mixture? And as a follow-up, were these human genes or feline genes?
We used human genes and mouse genes.
Okay.
Yes, in this study.
And did you try using less factors? Or was that the first kind of composition of factors that you
we
tried?
haven't yet tried, so we have to try in the future study.
Awesome job. I was just wondering what the efficiency was when generating these iPSCs. I know when you use virus-free vectors, it's very hard to get a high efficiency. And if so, if it was low, what do you plan to do to increase the efficiency?
Sorry, could you say that again slowly, please?
Yeah, what was the efficiency for generating the iPSCs with the episomal vectors? And what can you do in the future to improve the efficiency?
Efficiency, so maybe to up efficiency, so self-medium condition is important thing to up efficiency of reprogramming. So we have to modify medium condition to make feline iPS cells.
All right. Thank you so much.
Thank you so much.